rhymed with all the diseases you're going to hear about, or you have Jesaitis. It's a Lithuanian name. It's common ending. Lithuanian. I'm a professor emeritus. That means that the university has recognized me, but I'm retired, as you can see very happily, with my sunglasses and shades. But being emeritus also means that I'm allowed to still do work in my lab. The university is still giving me a lab. And I'm trying to finish up some things that I started, hopefully with a good ending. I've been involved in research for more than 40 years. I started here at MSU in 1989, coming from the Scripps Clinic and Research Foundation, where I spent 10 years establishing a couple of programs that studied the molecular basis of the inflammatory responses of certain white blood cells called neutrophils. And that's what I'm going to try to convey to you today and tell you a little bit about neutrophils, how they might relate to inflammatory bowel disease. So with that, let's start. And I've got to get used to having all these screens in front of me here. Okay. So if you look at a recent New England Journal of Medicine, 2009, it defines inflammatory bowel disease as an inappropriate inflammatory response to intestinal microbes in genetically susceptible individuals. And this definition, or this description of disease of this state, actually allows me to present just a very brief summary of how research has been organized in this particular area. So I feel very fortunate that after me, my colleague Seth Walk over there, who's also at Montana State, he's not emeritus, but he's also not retired, so he's actually continuing his work, and he will tell you about the GI biome. He's doing a lot of very interesting work on how microbes of the gastrointestinal tract figure in your health, and tell you something about the hygiene hypothesis. I think I'm going to have to use the old school methods here. Identifying now, see, I can't figure out which side I should go to, but the hygiene hypothesis, which you've heard a little bit about, and Healman for worm therapy that you've also heard about, which offer great promise. The other aspect of inflammatory bowel disease is this genetic component, genetic susceptibility. And I don't really do any of this, but over the last years, with all the omics that are being done, genomics, and the identification of different genes, and then the identification of variations in those genes, there's been an amazing amount of work that's been done to be able to correlate how those variations figure into various diseases. And 3 groups of genes have been identified that are associated with processes that result or that give the susceptibility to for inflammatory bowel disease, including certain genes. Of course, immunologists always and geneticists always pick the hardest words to pronounce and the most difficult way to abbreviate them, so nobody can remember them, and you have to sit and study them for days. I'm not going to really get into this, but suffice it to say that these groups of genes are now figured very importantly, and we have identified genes, their processes, and how we can study them to really get at the bottom of this genetic susceptibility. My particular area would be the inappropriate inflammation, and that involves the immunology that you've heard about from Dr. Schoenfeld, and where the immune system interacts with the blood cells to, in fact, result in these activation events And what's the net result from all this, from the therapy point of view, has been the identification using computational drug design. So the kinds of things that I do is I look and try to find what molecules are involved in a particular process, you get the structure of those molecules. From the structure, you can actually try to figure out what can fit into this molecule to make it not work right. Sort of adjust how these various processes and various pathways take place. There's the targeted immunotherapy that you heard about, and we've developed probes using much of the same methods that now are used to develop therapeutics that affect individually identified components of the immune response and the inflammatory response. And then finally, mitigation of this inflammatory response by the development of drugs and these therapies to really affect the autoimmunity. In fact, that's taking place when these processes happen in IBD. So one interesting slide, looking on the web, preparing for this kind of talk, from the standpoint of autoimmunity, IBD is in fact related to a number of different conditions that now are grouped into this autoimmune system. And what I've done here is circle Remicade as a target for TNF. This is, a therapy for the TNF target, which controls TNF, this cytokine that you heard about earlier. And if you can see that there are different pathways that identify other points of common function that really impact IBD. So learning about IBD, we also learn about these other autoimmune diseases. And all of them represent, in their final phases, some sort of inflammation that takes place. So in terms of the white blood cells that I've been studying, this is sort of this impossibly complex network that exists between individual cytokines that are produced by a variety of cells. Now, what's this cytokine? It's a protein molecule that's produced by cells that then is used as a messenger to tell other cells what to do or tell itself what to do. So they in fact are produced by immune cells, body cells, and tell the various cells what the state is around them. So they're kind of chemical messengers for immune cells. So many of these messengers signal inflammation, and we talked about inflammation, but what is this inflammation? Well, for 2,000 years, we've known that inflammation involves redness, heat, swelling, pain, and ultimate loss of function. If you look it up on Wikipedia, this is the image that you get. It's a condition called chilblains, where exposure of the extremities to cold results in an inflammatory response, and you get swelling, redness, heat, and pain. Now, for the pathologist, for someone who's studying the cells in the body, inflammation means something else, and it involves looking for a certain kind of blood cell called neutrophil or polymorphonuclear leukocyte. Now, all of you have seen these cells. All of you have had high school biology, and you've all seen blood smears. And this is the most abundant white blood cell in the blood. And it is an amazing cell that's basically used as a sentinel to look for points of infection and damage within the body. So in the blood, when it's circulating around, it just looks like golf balls, like many of the other white blood cells, in contrast to the red cells that you see here. There are about 5,000 of these per cubic millimeter of blood. Now, in the blood system, in the vasculature, the way the white cells are carried around are by the blood flow. And if I had a movie of this, and I saw some beautiful movies last week of this process, kind of wish I had them. But all the red cells would be flowing by here really fast. You could hardly even see them. And carried along with them are most of the white cells. But then, if you look along the edge of the vasculature, along the vessels, you see these cells actually rolling. Okay, they're rolling along, and these are the lymphocytes and the neutrophils rolling along, trying to find places where they should do their business. And they come across an area of inflammation where there's bacterial infection, or where there's like what you could call a sterile infection. You hit your hand with a hammer and you cause damage. There's no infection there, but the neutrophils know it. And they find these sites of damage or infection by spreading out on the vasculature and then crawling through in between these cells, they're called endothelial cells, through the tissue cells, and eventually arriving at the bacterial or damaged site. Okay, now this is a protective response of neutrophils, protecting us from, I'll switch over here for a little while, for infection that can be bacterial or fungal. It results in immune activation and also injury, as I said. So a sterile, kind of a sterile injury that results from trauma, from oxygen starvation. So what are some examples of this? So if you take a mouse and you inject fungal spores, aspergillus, bread mold spores, into the mouse's lung and wait a little while and assay the fluid that might accumulate there, you will see these enormous aggregates of neutrophils. They're primarily neutrophils, and in the middle of these are all these little fungal spores that are basically out of commission. The mice just take this as a matter of stride, even though you've thrown billions of conidia into these fungi. If you have an immune-compromised mouse, and this mouse is genetically deficient in one of the post-defensive processes that the white blood cell, the neutrophil, uses. Neutrophil is capable of making something called superoxide, which then is converted to hydrogen peroxide, which then is converted to bleach of all things. And that bleach is one of the agents that's used to kill these fungi and prevent this from ever happening. Because these particular mice are deficient in that they don't have the molecule that's needed to make that bleach. So what happens is you get these aggregates now that can't do anything. All right. Now, in a sterile injury, and a good case of that is a heart attack, let's say, you'll have uninvaded tissue and invaded tissue. This is tissue that's invaded with these neutrophils. Each of these black dots in here is a neutrophil. And they're accumulating these massive numbers because they've been signaled to go there by the damage induced by the oxygen starvation of the heart. And so when they accumulate, they unleash their microbicidal potential. That is, they start making superoxide, they start making hydrogen peroxide, myeloperoxidase, all kinds of oxidants. They're full of bags of little enzymes that degrade all the material that your cells have made of. So they're extremely damaging. And that's where the inflammatory damage actually arises from in a heart attack. You lose some function from actual cells that were oxygen starved. But if you deplete mice of their neutrophils, or rabbits or other animals, and you give them this artificial heart attack, the damage is actually minimized. So the neutrophils, the white blood cells, are the ones that are contributing in a major way to the damage. And the same thing is going on in your gut. So remember that, and this is a schematic of the gastrointestinal, let's say the small intestine, where you have microvilli that are highly vascularized, that is blood vessels going through, and they have this very thin layer right here, okay, of epithelial cells along each of these villi. But that's a very tight layer, tight, not allowing any of the bacterial products to get through. This environment here is just tightly packed with bacteria. We know, for instance, and it's been quantitated, that certain bacterial products that we know activate the neutrophils are here in concentrations that are enormous and would activate these cells. But when everything's functioning properly, the neutrophils just don't know it. And they'll just flow along through the blood cells. However, if there's a break, if there's some trauma, if there is an inappropriate production of these cytokines that you've heard about earlier, that will cause the neutrophils to stick to the vessels and migrate through and result in these kinds of cysts and abscesses where they accumulate in enormous numbers, releasing all their products, causing all this damage to your gastrointestinal tract. And this is basically the primary damaging process that's going to be occurring in inflammatory bowel disease. And ultimately, you get to the point where the abscesses get so big that they're even visible naked eye, as you can see, like the pus and lacerations in the gut, ulcerative colitis. So let's hope that this works now. So to study these cells in the laboratory, we use little pieces of bacteria called F-metluthi. They're pieces of protein. And what happens is we can have a little region that's embedded with this F-metluthi, and these are all neutrophils. And what's happening is the F-metluthi leaches out, and these little amoebae, which are neutrophils, are accumulating at this site. So the same thing would happen if we packed bacteria here, or if we packed dead cells here and broke them up with a laser or smash them in some way. The neutrophils would accumulate and start to be activated. And one of the activation that occurs here. You can see this beautifully as this neutrophil approaches a fungal cell. It starts wrapping its pseudopods around this and engulfing this cell. And what you see, all this red stuff here, that's the accumulation of a dye called nitroglyctetrazolium, which represents the generation of this hydrogen peroxide and hypochlorous acid, or bleach. And this is what kills bacteria. So we study both of these processes. We study the ability of the neutrophils to detect this F-metluthi, and we also study this process of superoxide production. We've purified components of these white cells that are responsible for both of these processes. So I'm going to talk about the bacterial sniffing response that the neutrophils have. This is what F-metluthi is. It's the N-terminus of these protein molecules, 3 amino acids. And it's very potent, activates neutrophils directly by a specialized protein called the receptor. And it's released by bacteria during an infection or when they break. And it's also released by cells. So where's it coming from cells? Cells don't make F-metluthi. Bacteria make it. Well, it turns out that a component of cells, mitochondria, is evolutionarily related to bacteria. Okay? And they still make formulated methional proteins, which F-methyl would represent. And the neutrophils actually recognize that as a signal for damage. And so they've evolved to recognize bacteria, but they also recognize dead cells. This is my old dog, Chester, a Newfoundland. And he has this amazing nose with a giant head that can recognize amazing food in any corner, anywhere in the house. So the dogs are able to sniff various odorants using cells, in fact, that have F-methyl receptors too. There's odorant receptors for F-methyl in the nose. And the sniffing, I call this the sniffing response of the neutrophil because it's able to detect bacterial fragments and broken body cells. This is a three-dimensional view, a model actually that we made of one of these receptor molecules. And it sits in the membrane of neutrophils. This would be the outside of the cell, and this is the inside of the cell. In my work, you know, I can't go into the individual things, but we've identified where in this molecule this F-methyl binds in here. We've identified what other molecules interact with this receptor and where it interacts and how it interacts with those molecules. The structure of these has not been determined to chemical preciseness with crystallography, but now many receptors, they have the same kind of structure and we can model them. Did you have a question?
Neutrophils have the ability to eradicate bacteria intracellularly?
Intracellular. They engulf the bacteria and they digest it. Most bacteria.
Pac-Man.
Yeah, most bacteria. And they
So not just the outside of the cell, they do actually go into the cell?
They don't go into the cell, they eat the cell.
Oh,
They take it
okay.
inside and digest it and kill it. So they're little amoebae that find a bacterium. They're about maybe 10 times the size of a bacterium in volume, probably. And they're actually capable of getting several at once and ingesting it and killing it. Now, that doesn't happen with all bacteria, but many of them. And there's a big process that's involved with so-called opsonizing the bacteria, allowing them to be recognized by and bound by the neutrophil so that they are activated as well. So this whole business, and this is a thing you can pick up on the web. It shows you sort of a schematic, very inaccurate schematic. I liked it because it has a pretty picture of our receptor right here with the F-methyl on this side and interacting with intracellular proteins, and then this complex array of pathways that has multiple effects that include activation of inflammatory pathways for release of cytokines and activation of the superoxide generating system. Lots of different things that are activated by the neutrophil. So our work in the most recent days involved trying to see if we could detect the state of these receptors when they're actually in a lesion that we have been able to isolate from humans having inflammatory bowel disease. And we use a process called immunofluorescence, where we've made synthetic laboratory-made antibodies. They're little protein molecules that we can tag with fluorescent probes, and when you shine laser light on them, they light up in a green color. And it turns out, so in this picture is a schematic that I showed before of the microvilli in your intestine, and if you make a cut right here perpendicular to these structures called crypts, you can then look at them face on, looking down. And that's what this next slide is. This is normal human colonic mucosa, and you can see the crypts here. And what's interesting, and this is a discovery that we made with collaborators, that these crypt cells, in fact, have the same receptor I just talked about that's on neutrophils. We didn't know that maybe 5 or 10 years ago. We didn't know that. This is a new discovery. And now these receptors are found in different places, including those cells. But they exist here, and they promote the healing of wounds. So if you have a wound in your gut, the receptors here recognize bacterial products, and they close up more quickly. Also, when we looked for neutrophils, which is what the money for this work came from the Crohn's and Colitis Foundation, we found in patients biopsy or actual surgical resection of patients who have ulcerative colitis, in this case it was a mild ulcerative colitis, we found neutrophils surrounding these crypts. And now notice two things. One, normally there are no neutrophils here. There weren't in the one before. And also notice that these epithelial cells are not being lit up anymore. So their formal peptide receptors are not recognizable in this case. They're not being expressed, and they're accumulating these neutrophils. In this area, it's called the lamina propria, the tissue surrounding the crypts. At high mag, here are the neutrophils. You never find them in crypts in a healthy person. But here in this Crohn's disease patient, you find them getting through into the crypt. Yeah?
Yeah, so you're saying that basically in normal villi they have these surface receptors that are normally present that will help to identify whether bacteria are present. Then when the neutrophils move in, those villi, the mucosal cells lose that surface.
They lose that expression of FDR.
So therefore, the white blood cells aren't going to go and destroy the mucosal cells. Is that correct?
No, well, the mucosal cells are operating as an independent system.
Okay.
The white cells invade the crypts and start doing their business and start killing everything inside. They start releasing superoxide, their degradative enzymes, and what we notice here that the receptors in the mucosal cells somehow disappear. For why? We don't know. I mean, they're regulated. And the neutrophils are here now, and this is just really a first shot picture. No one's ever seen these cells or this receptor in this kind of situation.
But the implication is that mucosa is damaged, it's not going to be able to heal itself.
Right, right. And the experiments that were done in model systems where you took a layer of cells, a certain kind of cells, and you made a mechanical scar in it, separated the cells, killed everything in between. Normally that would slowly grow together. If you throw methylphe in there, this little bacterial peptide, they start growing together 4 or 5 times faster. So the idea is that in normal recovery from injury, these cells are going to increase their recovery rates. But in this damaged situation, they probably are not doing that.
You're saying the neutrophils are not helping?
The neutrophils are not helping. The neutrophils are responding to all of a sudden the flood of bacterial products that are coming through the epithelial tight junctions and broken epithelium. And ultimately, they start piling up on one another and form these aggregates, much like the ones I showed you in the lung surrounding the fungi. Here they're forming these aggregates, and sort of the take-home message from that particular study, and it's coming out in the next month in the journal Leukocyte Biology, is that we have a way of protecting the unactive state and the active state of this peptide receptor. And we found that in these aggregates, everybody really considered this kind of the residual of dead neutrophils and dead cells that accumulate into this pussy abscess. But our finding, and needs to be confirmed, is that the cells on the surface, the neutrophils on the surface here, still have active receptor that's not been shut off and may still be contributing to the activation of more cells coming in there and propagating the inflammation. So, so that's kind of a view of the small amount of research that we've done. You have to consider that there are many receptors in the neutrophil that stimulate similar processes. There are receptors for all the cytokines. All of these things are acting at once. And there's this incredibly complex network that has to be sort of figured out. And really, I mean, I'm at the end of my career. I'm not ever going to figure this stuff out, but somebody with some really good computer skills will be able to come in doing these omics studies to be able to identify how we can get a handle on this disease by attacking a specific component somewhere, some magic bullet maybe, like, you know, like infliximab or the Remicade that we spoke about before with the TNF. So in any case, I thought I'd give you an idea. It leads to other questions that we have about this, but ultimately it just requires hard work getting in there and a bunch of people studying this stuff. So I wanted to, again, thank Kathy.
And money, too.
And money, right, money. And that's why I want to thank Crohn's and Colitis Foundation for this work. National Institutes of Health, I'm bragging, but I have I hold the record at NSU for the longest running NIH grant. You have to renew them every 5 years, so I had a good run. And then Montana State for giving me my lab and giving me the opportunity to do this work. And also the clinical lab at Bozeman Deaconess, because over the years they've provided us with fresh human normal blood that we study neutrophils with, and many students, colleagues, and collaborators here in Montana and all over the place. So thanks very much.